I guess there's enough here. I'll go ahead and start for the day. So, as the title suggests, we're going to start at least observing real data of these FASTQ files that are going to hold and relate to RNA-seq data. So FastQs can hold anything, like anything that comes off an Illumina is going to be a FastQ. A lot of sequences, even the third generation sequences, are going to be displayed as a FastQ. So there's really a lot of this stuff is universal, regardless if we're talking about FastQ or RNA-seq or whole genome sequence data. The things is going to be the same. There's going to be a lot of overlap. I just like using RNA-seq data because it's usually smaller than genomic data, and it's going to hold a lot of. I think I can do more stuff with it easier than I can with whole genomic data. Plus, it's smaller, and so we can effectively do the same things, and it's going to be faster and take up fewer resources to perform. So for y'all that are in this class, I've put in some example FastQ data for you. That's in the data directory, and so it's this ERR numbers.fastq. And so this is available to you. You can copy it and put it into your own directory, and this is just for testing stuff and playing around with. I don't know if y'all can hear the siren, but like every day or every Wednesday at noon, aka during my class, there's always a tornado siren, just a test siren. It's annoying. I don't know if y'all can hear it or not, but it's super annoying. It seems to last forever, right? It's been 3 minutes on. Anyways, so these ERR numbers. And to recognize these files aren't exactly small. Each of them are 2 gigabases apiece. And again, so instead of seeing an R1 or an R2, that's going to be denoted with an _1 and an _2. Principles are going to be the same, right? We have basically a left-hand file and a right-hand file, or forward direction, reverse direction. But it's just going to be noted with an _1 and an _2. So go ahead, and if you're here, if you're playing around, or whenever you want to, you can copy this into your home directory, so we can play around with it, take a look at it, and see some properties with it. So I've already done that. It's in my test directory, and specifically I made a new directory called FastQ that has those ERR numbers. It has some other stuff in here that I'll talk about in a second. But notice, here are the main files. ERR_1, fastq.gz, ERR_2.fastq.gz. And so these FastQ files, you do want to keep them as a gzipped. Because unzipped, these files get massive. So almost always keep your FastQs as a gzip. Again, we can manipulate them, we can look inside of them, we can read them with using zcat, right? So zcat lets us at least print a few lines to the screen. Specifically, if we do a zcat ERR_1 and pipe that to head. Because if you just zcat this, it's going to print the entire file. It's going to take forever. I mean, it's 2 gigabytes long, zipped. So it's just going to take forever to scan through the whole thing, plus all of it's not always necessary. Just the first few lines are all that we need to look at to see what's happening in this FastQ file. Five minutes in, siren's still going. Anyways, so now just for reference, these libraries, these ERR libraries, are derived from a human testis. If you know anything about me or know anything about my research, the only organ that matters to me is the testis. It's a weird tissue. It's always in meiosis. Does a lot of weird things, and it's my tissue. So these libraries are from a human, from human testis, and pretty much all RNA expressed in this sample. I kind of stole this from a student because he already had a file ready to download it, so I just went ahead and re-downloaded it and made it available for everybody. Just again, this is just for example purposes. So I'm going to go ahead and bring that head up just to demonstrate what we've already talked about on Monday, right? So each sequence is going to start with an @ sign. There's going to be the name of the sequence, some attributes about it. So what this says is it came specifically off of a HiSeq Illumina machine. Commonly today it's like the most popular machines used are going to be a HiSeq or a MySeq. And this is specifically from a HiSeq that's denoted in here. And it tells me that the reads are 100 base pairs long. 101, but you know, kind of 100. And so each name, right, each read is going to have a different name, right? So ERR library 0.1. And so again, it's going to be 4 lines long. So we have the name, then we have a sequence. And then, so I mentioned that normally it's a plus sign, like the third line would just be a plus sign. Older FastQ files are going to have the name repeated. Newer ones won't because it saves space. So a lot of it's just a file. It's just a preference. Nothing specific. It's just kind of an older-ish format. And then this last line is the quality scores. And again, we can't actually read the quality scores. Right? It's symbolized. We could, you know, figure it out what specific number this F refers to, but it's going to refer to a quality score. And so all of these lines refer to the first sequence. And then the second sequence, all of the next 4 lines are going to describe the second sequence in this file, etc., 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 for miles and miles long. And the same thing is going to be in the _2 file, right? I mean, it's going to be the exact same stuff. And again, the nice thing about it is that these reads are paired, right? So the first read of the second file is going to be the pair of the first read in the first file, right? So everything's paired, and again, it tells us the same information. 4 reads per sequence, things those kind of attributes. So now we can summarize this entire file. We can basically summarize the quality of each of these files. You know, how good the reads are overall, right? We can't look at every single read. We certainly can't interpret the quality scores, but we can let a program tell us more about this library. And so that program is FastQC. FastQC. And so FastQC is great for just visually examining the quality and the properties of the sequences in these files, right? So it's going to summarize the entire file and basically summarize everything that's there. And so FastQC is really easy to use. All it is going to, you can do both files at once. We can do err_1.fastq.gz and err2.fastq.gz, and both will be analyzed and processed. And so I've already done that, and that's what all of these extra files are, because this does take a little bit of time to run. And it's just because we're running this program on 4 gigabytes worth of data. It just takes a long time to process that much data, right? So I don't want to spend a whole lot of time just watching a program run. Feel free to do this on your own. What's ultimately going to happen, and I can at least show you what's going to happen. Let me make a quick directory so none of my stuff gets overwritten. Oh, come on. Come on. Make dir mkdir. There we go. FastQC. Cool. Okay. Right, just so we can see what happens when this stuff does run. Make sure so you know what to expect. FastQC. Right, it's just going to start the analysis and then go in 5% increments and just say 5% done processing, 10% done processing, 20% done processing, etc., etc., etc. So just so we can demonstrate that, right, 5% complete. And so this is just going to run until it goes to 100% complete, and then you get output files. And that's what's going to be here. Right, so you'll get 3 output files. You'll get a fastq.html, and a fastq.zip. I should say only 2 files, HTML and a .zip. The file of interest is going to be the .html, because we can look at this, right? An HTML file we can examine in a browser. So again, I've already copied this file over. So what you would have to do is bring this file, these HTMLs over using FileZilla, right? Use the FTP transfer, bring it over to your computer, and then open it in your favorite browser, right? You double click on it, it's going to open in your default browser. So I'm going to go ahead and do that real quick. I'm just going to look at the _1. So we can see what the output of FastQC looks like. All right, and so it tells us some attributes about the file. It will tell us how how these bases were called. It'll tell us how many sequences are actually present. I'm going to zoom in on this a little bit. Tell us how many reads were present. How many were flagged as poor quality, the overall length of each read, and what the percentage of a CG is. Okay, so that's one thing that it tells us the basic stats, and then it tells us the average base quality score per base among all of these millions of reads. How many reads are actually here? We have 32 million reads. So as well, that's not very many. Yeah, it's a great number for RNAseq data. I should say that 32 million is a pretty good number. So again, like FastQC already encodes, tells you like what's a poor quality read, what's a poor okay quality read, and what's good quality base calls. Right. So we've already talked about like this Fred scale. So these quality scores are for each base at position 1, 2, all the way to 101, or at least summarized. And so on average, the first base is called with a quality of 32, right? So anything above 28, FastQC is going to call a very high quality base call. Anything below 20 is considered meh. Because again, remember this is like out of a log scale, so with a Fred of 20, it's basically saying at least 1% of bases called with a Fred of 20, with this quality of 20, is called wrong. Now, I mean, we have millions of sequences in here, so even a 1% error rate is going to yield a lot of errors. And Fred of 30 is 1 of 1,000 are called wrong. So that's getting better. It's still, when we talk about millions of reads, millions of bases, right? 320 million bases, even with an error rate of 1 out of 1,000, 0.01 or 0.1%. That's still a fairly big number, right? There's going to be, even with a Fred of 30, there's going to be some miscalled bases in here. No surprises. But that's all we can do. We have to have the whole read. We have to take it into consideration and work with what we have. Now if we notice, right, they're basically box plots. It tells us where on average where most of the data lie, where most of the quality scores lie per base. And so I would say that this library is a very high quality library because most of the bases are called with a Fred over 28. Yay. So very, very good library. I've definitely seen some trash libraries and even made some trash libraries that at best case scenario have only a Fred above 20. No surprises. Work with what we have. So it's not uncommon, especially like prior to 2010, it was not uncommon to get rid of bases that were called with less than a Fred of 20, and that's kind of the threshold. A better threshold is 30. Anyways, now if we see a trend in this, is that the qualities will start off pretty high, but as we reach the 3' end of the sequence, the qualities drop off a little bit. Again, this is a good library, so it's not that dramatic, but nonetheless, the qualities will drop off at the 3' end, and that's just a product of the sequencing chemistry, right? Because how Illumina works is like we sequence one base at a time, and as a base is added, that base will glow a certain color, like it will produce a specific color of light that a camera will detect. And so this happens in a stepwise fashion. So we add a base, flash a light, stop everything, clean it up, add a base, flash a light, stop everything, clean everything up, add a base, etc., 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 for 100 rounds. But as we get deeper into the sequence, the flashes of light, remember this is all from a single cluster, and so if there might be a base missing in that cluster, like something had gone wrong in the PCR, the bridge PCR, right? There's always mistakes in a PCR, and so that cluster won't all emit the same light all at once, right? If it's supposed to all emit yellow for a specific base, it may not always. It might get it like a mix of yellow and blue, and so that kind of says like, well, we're struggling to call what this base is. And it's just the trend of the chemistry is that the quality decreases as we reach the 3' end. As they reach the tail, it just kind of drops off. Whatever, we can deal with it. Not important. Quality scores, again, this just tells us the average quality scores, and most of our bases are called with a score of about 36, 37. Really good. Really high quality reads, because this is like the average quality over the 100 bases per read, and so the average per read is solid. That's a good library. Base per base sequence content, right? You would expect ish that bases would all be in about the same percentage at about 25%, right? Each base should be present about a quarter of the time. But we see a little bit of wonkiness in the first, like the first 5 or 10 bases, right? There's a little bit of scatter. Again, it's just kind of some weirdness in the chemistry. It happens, and we can deal with it. And you kind of see that in the quality scores, like the first 5 or 6 reads, even the first 10 reads, they're not going to be as called as well as this middle sequence. So really, the best stuff is going to be from like base pair 10 to base pair 75 in this specific library. And what else? What else we can see? We can see CG content. Man, that's fine. Whatever. It is what it is. Not a big deal. Base in content. That's good. Sequence length. That's all the same. Sequence duplication letters or levels. It means like if the same sequence is repeated over and over and over again, that will be referenced here. And sometimes we'll see overrepresented sequences. Right, so that's going to be an overrepresented sequence in this library. So it repeats about 100,000 times. And the nice thing is that FastQC will recognize adapters. Right, so it's saying that this sequence is just part of an adapter. Okay, no problem, we can deal with it. And this is the last thing that's kind of interest, is this adapter content. And so I've drawn this out, and I'll demonstrate this in a second, but it's saying that there's a universal adapter, there's an Illumina-specific universal adapter, and it seems to be more common towards the 3' end of each sequence. It's not that common, right? So it's only in like 10% of reads, and only for like the last 40 bases. It's not that common. But how this is happening is I've drawn this out. So it shouldn't lag because I'm not going to draw it out. I've already drawn it out. But if we think about an actual sequence, what's happening whenever we sequence? So we have this RNA, so the cDNA, as I labeled in blue. So this is the stuff that we're interested in, is everything labeled in blue. Remember, so just part of the adapter, or sorry, these adapters are added during sequencing, like during the chemistry. As we're doing the library prep, these adapters are added, right? So this will be like an Illumina specific adapter, okay? It's just there, right? We ligate it on, it's there. And so our sequencing starts from this adapter. So if we're sequencing the forward strand, we start from the 3' end, and we'll have like a little primer-ish that will bind to this adapter region, and then start sequencing inwards. Okay, and so if this region is short, so remember we're sequencing 100 bases at a time, and so if this region, if this little fragment is less than 100 bases away from this 5' adapter, we will sequence a little bit of it. So we start sequencing here and move inward from 5 to 3. And so sometimes our 3' end will have a little bit of this Illumina adapter. And it will happen on both strands, right? Or it should happen on both strands. Not always, but sometimes it will. Right? So the same thing's going to happen in the paired, right? We start sequence all the way through, but sometimes we'll sequence into the adapter. And that's something we can deal with. So ultimately our final product read is going to have a little bit of the DNA of interest and then a little bit of an adapter. All stuff we can deal with. Right, so these reads, again, are pretty good. These reads are pretty good. That being said, I'm not going to spend a whole lot of time today on cleaning these reads up, because that kind of is a line that requires a little bit more explanation than what we're going to have to do today. So on Friday, I will demonstrate how we can clean this data up and make it look nice, right? It's already good data. It's like this is already usable in its current state, but we can make it a little bit better. We can trim off some of these adapters and remove some of these low base quality reads. So I'm going to leave it at that. I'll at least give you a chance to at least replicate this, right? Build this FastQC yourself, or build this FastQC output yourself and take a look at it. It should all be the same because we're all messing with the same library. Okay. Again, we'll deal with cleaning these reads up again on Friday. But before we go there, as I at least want to demonstrate where we get this data from. And so this data, this type of data, is stored in NCBI. So NCBI is like the national, it's hosted by the NIH, it's funded by the NIH, and it's the National Center for Biotechnology Information. But really, it's just a repository for all DNA sequences in really the US. Most of the time US. There are mirrors. So like this data is accessible, and also a European mirror, I think the ERA, and Japanese has a mirror as well. So they can have the same, they have access to the same information. And so NCBI, again, just holds all known sequencing data, pretty much all of it. If it's ever been generated, especially under like a government grant, that data is required to go here as a condition under that grant, right? If you get money from the government to do any biological research, any kind of bioinformatics, and generate this data, you have to deposit into a publicly available database. And 2 seconds. So you can access anything that you want. And so FASTQ data, like RNAseq data, is specifically held in a region of the NCBI. And so we'll explore a little bit more about this NCBI in a little bit. Like whenever we start identifying sequences, I'll kind of talk about BLAST a little bit. But really where we're looking is the SRA. And the SRA just stands for the Short Read Archive. I hate the SRA. I absolutely hate the SRA. I'm sorry, but I hate the SRA. And I know I'm not the only one. Because the SRA is a pain to navigate, and it's a pain to get data, and it's a bigger pain to upload data to the SRA. Hate the SRA. But nonetheless, any sequencing library that's been done is on the SRA. And just to answer a quick question, so this is different from single cell RNA sequencing because this is from a tissue, right? So we're isolating a full tissue and extracting RNA from that. We're not doing this from a single RNA-seq cell. But honestly, there's probably really no difference. You probably analyze it the same. I'm not a single cell RNA-seq guy. Honestly, I kind of think it's a waste of time and a little bit more expensive than it really should be. Sorry. Right, but this is from a tissue, right? We just isolate RNA from a tissue. And I say it's kind of a waste of time because the tissue seems to mirror a single cell most of the time. Anyways, nonetheless, nerds, there are a bunch of single cell RNA-seq nerds on the internet. I know, sorry. You do you with your data. Don't worry about it. All right. So with this SRA, I kind of hate the SRA because it is kind of a pain. But again, I can find anything that I want. Almost anything that I want is available here. So I can literally do like human testis. Right? I'm just typing in these words, human testis, and letting it search for any library that may have human testis. And so we can kind of further isolate the libraries that we would, right? So we get like 20 how many hits do we have? Like, about 3,500 hits. Right, 3,500 libraries seem to be somewhat related to human testis. So crazy. And so like we can pick whatever we want. But for example, I don't want DNA, right? I don't want DNA. It doesn't matter what cell we get DNA from. It's all going to be the same. RNA, right? We want RNA. So I can click on RNA to filter out all the non-DNA libraries that I care about. Further, I can also just get the paired libraries because I don't like really working with single end. It's kind of an older technology, not as helpful. It's half the data, so I filter for the paired as well. Blah. So then, right, we're still limited to like 2,100 libraries that we can all filter through, try to look through. It's 109 pages. I'm not going to do that. Right, so there's like the annoying thing about the SRA is like I specifically look for human testis, and it still gives me results for mouse, ratus, that's a macaque, that's a primate, and other things. Kind of annoying, but whatever. Nonetheless, this library is searchable. So the library that I've given you is searchable. This ERR number is going to tell me or tell you where this library come from. So if I just search for this specific library, it should give me all the information about that library. Okay, there it is. So in this it tells me how the library was constructed. Now this varies from library to library. It just depends on how thorough the researcher wants to be. Because as you load it up, you try to provide as much information as you can, like how this library was prepped, what species this is from, what tissue this is from, is it published? If it is published, what paper lists it, things like that. Okay, tells us how big the file is, how many bases are in it. This is like the number of reads, how many bases, right? 6.5 billion bases in all of this, how big these files are, and when it was published, right? As I said, this is kind of older. This ERR kind of denotes an older library, so 2013 is kind of older. And if we click on the ERR number, it's going to give us some more additional information about this library. Right, tells us it's all paired in, blah, some of it's 95, yeah, so they did RNA sequencing on 95 individuals from 27 tissues, basically to find a bunch of coding sequences in the human genome, right? That's how that's kind of done. So if you want to know what every single gene is in a genome, you kind of have to get every single tissue, RNA-seq it, and go from there. Anyways, now so this is another thing that I'll deal with on Friday, but we can download this data. So downloading stuff from the SRA, huge pain. Not gonna lie, it's kind of a huge pain. I should say it's a huge pain figuring it out, how to download it. But how we can download this is by using the specific ERR number, right? So not this, there are a whole bunch of accessions, like we have this ERX number. The most important one is the ERR number. Sometimes it's an SRR number. But nonetheless, we can copy this, and it's going to be kind of similar to like wget, and it's going to be kind of similar to git. But here, I'm not going to run it, and if you try to run it, you're going to run across an error. So I would encourage you not to run this line, because we have to initialize a program before we can actually run this command. But whenever we do, it's going to be fastq dump, and then the ERR number. Right, and that's it. That's all you have to do. Even further, it's nicer to make sure that it's gzipped and split. Oh gosh, I think it's split read. I think it's split read. Yeah, there's a gzip option because we want to keep it compressed to save space as we download. Keep it spaced, and yeah, split files. And so since this is a paired in read, if we just downloaded it by itself, both of the reads are going to be stitched together kind of like this and downloaded as a single read. But we don't want that. We want to split them apart into left and right hand files, or at least an _1 and an _2 file. So if we ever run fastq dump, it's going to be with the --gzip and split files, and then the ERR number. Oh, come on. I need ERR number. I need that ERR number back. Okay. Right, and so running that, it's going to take forever. So again, if you try to run this yourself, or if you're in this class and you try to run this yourself, you're going to come across a stupid error that we'll deal with. But that's effectively how we're going to download these libraries. That's how we can get them. Anyways, one thing I can show you with our remaining 10 minutes is something that helps us have fewer problems. So downloading 4 gigabytes of data is going to take almost an hour to do. It takes a long time to download that much data. And we're all kind of on a VPN. We're connected to the server. We're not actually on this server. We're just sending commands to this server. And so if we get any kind of internet hang-up or hiccup, that process will stop and we lose our download. Huge pain. Huge pain. Done it, lost data, had to restart, I don't know how many times. Huge pain. But what I can talk about today, with our remaining time, is how we can avoid that problem. Avoid hangups when we have long things to run. Because whenever we run our RNA-seq data, it's just so much. It just takes a long time to process. It takes a long time for FastQC. It takes a long time to clean it up. It takes a long time to analyze it. So if we want to avoid hangups, we can use a very semi-user-friendly program called the Tmux. Tmux. And so Tmux is awesome. And unfortunately, I probably learned Tmux like 2 years into my bioinformatics informal education. And I wish I had started from the very beginning, because Tmux is awesome. So whenever I run Tmux, what's going to happen is I'm basically opening up a program on the server, right? So instead of like sending commands to the server, Tmux is going to emulate like I am sitting at that computer running commands directly on it. And so whenever I actually start Tmux, what's going to happen is there's going to be a green bar that opens up on the bottom, right? So now I've run Tmux. So I have this green bar at the bottom, and it has zero bash. And so what's happening now is it's like I'm sending commands to Tmux. Tmux is going to run those commands on the terminal. So now it's like I am sending commands. Now it's like I'm more or less sitting at the server itself running commands. And so I'm going to send out a little cheat sheet to help with Tmux, because it is kind of tricky, because we can do a lot with Tmux. And I'll demonstrate a little bit of that. But before really demonstrating anything, Tmux requires something called a prefix. And the default prefix is control plus B, control B. And so like anytime that we want to manipulate the Tmux shell, we have to initialize it with this control B. So one trick that I'll kind of immediately do is I'm going to split this screen. And I'm going to split it in half vertically by doing control B, then the percent sign. Okay, so I'm just going to hit control B, and then the percent. And so now Tmux is split, right? So I have one terminal reading one half, and then I have another terminal, and both of these are independent, right? It's effectively like I'm running two terminals at once. And so another command, so if I want to switch between these, it's control plus B, and then O, lowercase o. O. So if I do control B, O, now I'm working over here. If I get control B over here, then I'm on the right hand side. So it's like things are happening. I don't want, okay. Where am I at? So I can head this ERR file here, zcat head. And I can do something over here, just with Tmux, and that's kind of awesome. Furthermore, I'm going to run this command called sleep. And so what sleep does is it's like do nothing for a set number of time. So if I do sleep, say sleep 10, I'll do sleep 5 to just demonstrate this. So if I do sleep 5, it's just gonna pause for 5 seconds and then return back to the terminal. So it's not really doing anything, it's just sleeping. Okay. And so this last command that I'll demonstrate is a detach, because right now I'm attached to this Tmux shell, but I want to detach before I want to log off of the server, because I can leave a command running in Tmux, log off of the server, come back, and that command will still be running. Awesome. And so this detach is going to be control plus B, and then D. So I'm going to demonstrate that I can leave a command running, detach, and then come back to it. So I'm going to sleep for 60. That's 60 seconds, right? So I'm just going to sleep for a minute. Control B, D to detach, right? So now it says I have detached from session 4. So that means I have I think I have 4 Tmux shells running. Whoops, that's my bad. I can fix that. So if I want to see all of the sessions that are currently running, it's going to be Tmux list sessions. I only have 2 running. Okay. So I have Tmux 1 and Tmux 4. So these are just the names of these Tmux shells that are currently running. And so I can return to those shells. So I'm going to return, so I know that I just detached from session 4, so I'm going to reattach to this. And so that command is Tmux attach -t and then the name. In this case, it's going to be 4 because that's the name. And so now I'm back to that state, and my sleep is still running. Like, my sleep is still running. Right? So whenever we start doing FastQ dumping, downloading files, running these very, very long commands that just take a long time to process, you want to run it in this Tmux shell. Right now it's done. Right? It finished. Right? Sleep 60 finished. So I can run something in Tmux, detach, come back to it, log off, log back on, reattach to my Tmux, and everything will still be in place as I left it. Hugely awesome. And so if I want to close Tmux in general, it's just going to be Control D. It's just Control plus D until everything closes. Right? That's going to be exit. Right? So there's a huge difference between detaching and exiting. Detaching means like I'm going to leave the Tmux shell so I can do other things, leave it running, and come back to it by attaching to it. Alternatively, this Control D means close it all down. Right? So I'm just I Control D to close that window, I Control D to close that window, and now I'm exited out. That Tmux server, that Tmux shell is going. So if I Tmux list sessions again, it's just going to tell me that Tmux 1 is running. And so this is old, right? So I started that on March 9th. Doesn't necessarily mean I had a command running, but if I attach to it, I'm kind of nervous what I have here, -T1. I have nothing going. I demonstrated this for another student earlier. Tmux attach. And so all students in the class have access to Tmux. I'll send out a cheat sheet that help mess around with it. So like, for example, I can also have multiple windows with Control B, then C will give me a new window. So Control B, C. Crap. Control B, C. Right now it made me a new window. So you can kind of see that happening. If we look down at the bottom, we have this 0 dash. So let me just run that again. Control B, C, and now I have another window. This other new window within my whole Tmux shell. And so I can do the same thing. So I can have something running and use Control B in to move back to the original window. So that's Control plus B, then in to switch windows. So right, Control B in, it's still sleeping. Control B in, I'm back to the original. Control B in, still sleeping. So I'm just going to kill that. Again, to close this window, to close every window is just Control D until it can't, and we're done. We're exiting. So now I should have no more servers running. I have no more Tmux sessions running. All right. And so on Friday, we'll play around with cleaning these reads up, right? Because that's going to take a little bit of time in itself just to explain. So until Friday, I'll see y'all later.